This is the Embryology Virtual Lounge from Fujifilm Urban Scientific. This is Bingu, the Technical Application Manager from the company. Today's topic is about MEGA. It's the mouse embryo genetic assay developed by our company. So it's mostly about the QC for the field. And before we started, I want everybody to know that, first of all, I thank you for joining. And the whole session will be recorded just in case everybody else who cannot attend this session today, we can share with them the link and about the video. So yeah, let's start our session today. So as I mentioned, it is the topic today is called MEGA. So the question is, what is it? Let's see. So it's about quality control. Okay, so in this field, no matter for the equipment, for the labware, and for media, before all the product came out of company, we need to do quality control. So normally for media, before we send it out, we will take we will measure the pH, test, check the appearance of media. We will do the sterility check, do endotoxin check, and osmolarity check. So, but what about the function? Normally, as we know, it is common sense that all the media need to take MEA test. What is MEA? MEA is mouse embryo assay. And here's some picture about mouse embryos. So normally the test will be done like, we will start to culture the mouse embryo from one cell and culture it for 96 hours. And during the 96 hours, it will develop into blastocyst. So in this assay, only morphology is checked. And the criteria is if only 80% of the embryos form blasts, the test passes. However, there's still some limit to MEA. For example, we always joking that if you just put the mouse embryos into tap water, it can still grow into blastocyst, which means it is not that sensitive to the toxicity or suboptimal culture environment. And also, we have the common sense that the mouse strain is very important for the sensitivity of the assay. So for this part, we're looking, while looking into the future, we develop MEGA, the genetic mouse embryo assay. So here is two pictures about MEGA. You can see on the right side, it is a picture of a mouse embryo at 48 hours. It is a normal morphology check. But for MEGA, and under the fluorescent scope, you will see it will have green fluorescent, and the blue one shows the nuclear. So that's what you will see from MEGA assay. So here is a summary about MEGA. So for MEGA, we use this genetically engineered mouse strain. And we also use this fluorescent detection of a developmental regulated gene expression. At 48 hours, EFI, which stands for early fluorescence intensity, is checked. And at 96 hours, fluorescence localization is checked. And by all means, morphology is checked at both times. So the specifics for MEGA is that we are using this specific transgenic mice, strain B6. And the gene CBA-TG, also named as POU5F1 EGFP, is randomly integrated transgene that expresses enhanced GFP under control of the POU5F1, which also have the name of ACT4 promoter. So it is actually the embryo will express biomarker-linked fluorescent tracer protein. So that is, we will, by detecting the green fluorescence protein, which shows green light, and we can see the ACT4 expression. So here is a picture showing how GFP is inserted into the protein. So normally, it's the protein without, you cannot see it under the scope. But while we just inserted the GFP as the tracer into the protein, it will show green light under the fluorescent scope. So here's the question, why we measure ACT4? So by many articles, ACT4 is a transcriptional regulator in oocyte and pre-implantation embryos, which can indicate normal development. ACT4 is closely associated with pluripotency, and the amount of the location of ACT4 GFP reflects normal development. The lower level of ACT4 expression means the failure of ICM formation, pluripotency loss, and differentiation to TE. So here is a paper from 2014. It shows that ACT4 is required for lineage priming in the developing inner cell mass of the mouse blastocyst. And yeah, so that is the part why we choose ACT4 as a indicator for this MEG assay. So here is the picture at 48 hours. So as 48 hours for mouth embryos, it normally will be, you see it's the cleavage stage. And under the fluorescent scope, you will see if it's the normal one, the plasmid will show green fluorescence. And as I mentioned, the blue color indicates the nucleation for each plasmid. And if the embryo is not that good and have less potential to develop into blastocyst, even though it will it still have the normal morphology, just as is shown on the right side. Under the fluorescent scope, you will see there's low expression of ACT4, just as shown on the right side. There's really little green light showing on the scope. You can only see the blue nuclear. And also on 96 hour times, the fluorescence localization will be checked. So normally the ACT4 should be located in the ICM. So just as shown on the right side, you will see for the full blastocyst, 3 have expression of ACT4 in ICM, and the one on the left corner, it almost have non-ACT4 expression. So which shows it have less potential to implant, or the quality of the embryo is not that good. So when we developed the genetic mouse embryo assay as MEGA, we did publish into in the Journal of Reproductive Biology and Endocrinology. So this is the article published by the director of R&D, Rebecca Gilbert. So if you guys who are interested into more details on that, you can just find the paper, or we can share with you. So here is more pictures that shows how MEGA works. So you can see still at two time points. The first series is about 48 hours. So for 48 hours, it's still at cleavage or morula stage for mouth embryos. So for those good embryos, it will have really strong green fluorescent under the scope. And for the one which have some potential to develop further or to be implanted, it will have less or weak green fluorescence. And at 96 hours, we will also check the localization. It should be normally mostly localized at ICM. So this is also, you can see on the left corner, the one half is poor in morphology as blastocyst, and it also shows less green fluorescent. So here's two charts which shows the EFI vs morphology. EFI stands for early fluorescence intensity. So it is the GFP expression at 48 hours. And from the data, we can see that EFI is more sensitive to suboptimal condition than morphology alone. And EFI at 48 hours demonstrates predictive value of 96 hours blastocyst rate and the viability via expression patterns, localization of GFP in ICM. So you can see the charts in this slides. So the one which show on 48, you can see the blue bar stands for the morphology, which have more than 8 cells at 48 hours. And the green one shows the fluorescence of high EFI. So you can see as in control group, control means the culture environment is perfect. So you can see the morphology at that time over 8 cells has about 65.9%. And at the same group, the high EFI shows 89.8 as percentage. And for the suboptimal in culture environment, you can see still the morphology is not that sensitive. It still have 63.3% to grow for more than 8 cells. But when check the high EFI, it only accounts for 33.3. So which means the EFI is more sensitive to suboptimal condition, and we just follow up that as there to 96 hours. So you can see that. So for the control group, that which is just control group for the which grow to expanded and or hatching blastocyst accounts for 86.4% in control, which means perfect culture environment. But for suboptimum group, the ones that can be grown to expanded or hatching blastocyst only accounts for 30. So as you see on 48 hours at the suboptimal, judging from morphology, it shows good, it shows okay. But judging by high EFI, it shows just the similar result like for the 96 hours. So it means it has, which demonstrate that it has predictive values of 96 hours blastocyst rate and viability. And this is some case study for Mega using for a protein screen. This is for HSA. So we did test like 5 batches of HSA raw material. So as you can see on 48 hours, all for morphology only, all 5 batches of raw material has reached over 95% of good morphology. But for ECI, the batch of A and D shows potential potentially suboptimal. So when we pre-culture the embryos to 96 hours, so you can see the ones that has grown to expanded and hatching in morphology H&A is only 72.7%, which means it hasn't passed the MEA. And for D, even though it's over 80%, but it's still low as just 83.7. So this means these, just as shown by EFI at 48 hours, these two lots have potentially suboptimum, but we can judge it from just at 48 hours. So the combination of EFI and morphology allows for selection of highest grade material. We also done some tests trying on the oil. So this is actually just an experiment. So we do with the literated oil, which is as control, and for both normal both the normal EFI and also the expanded blasts at 96 hours, it shows good. But you see, when we add in 5% of adulterated oil into the perfect one, for the morphology, for the expanded and Hatching blasts percent compared with control, it still works the same. So, which means if we just do the normal MEA, it will pass. But when we check at the 48-hour normal EFI, it shows only 63.1% half of the control. So it shows some concern of the raw material. And when we adding the bad oil into increasing to 10%, you can see both the EFI and the blast rate dropped. So it also shows how EFI is much more sensitive than normal MEA while judging by morphology. So here is this a summary of the comparison between MEGA and normal MEA. So as you can see, for normal mouth embryo assay MEA, we normally just graded the morphology at 96 hours. And for MEGA, we will check the EFI at 48 hours, just as gene expression. And then we will also observe the morphology at 48 hours. And for 96, we will grade the morphology as always, and we also will check the gene expression. So by combining the morphology and gene expression, we can, as a QC method, MEGA is much more effective than normal MEA. So right now, we're just doing it with our board, using it to test the raw materials. So which is only including the protein, which is HSA and alpha beta globulin. And also we will test the raw material oil. So this will also transfers to all the complete media with protein supplemented. So our goal is to have the more rigorous QC method, to have the more sensitive assay, and for raw products most sustainable to contamination. And we want to improve the end products with a more sensitive and effective way of QC method. And we just by inserting a single gene, we want to have the whole field to as an increasing level of QC to provide a safe and stable environment for embryo culture. So this is MEGA. This is what we are talking about today about the QC system. And thank you. So do you have any questions? Yeah, so if you have any questions, you can type it in the chat boards, and also you can unmute yourself and ask by voice. Yeah, so besides the everything, as last time we were talking about the critical parameters in the lab, and today we're talking about the QC system in this field, and we are trying to improving the whole system to increasingly focus on the security and efficiency of the system. So that's why we're developing. So as a media pioneer in this field, we're trying to do the best to make everybody, the embryologists, feel at ease. Any questions or any opinions you want to show? Okay, it seems everyone is clear. Okay. I still haven't seen. Okay, so I got one question about, so we need fluorescence microscope instead of normal inverted microscope. Yeah, for mega, we didn't need that. Hello? Yes, for mega, if we need to perform mega as a QC methods, yes, we do need a fluorescent microscope instead of normal inverted microscope. So yeah, right now that's what we want to do, that's probably the equipment needed for this test. So any other questions? Okay, if there's no more questions on this topic, and I will call it a day. And thank you everyone to joining into today's session. And we will continuously to set up this kind of virtual launch to provide a platform for everybody to join in, to take a rest and think and to communicate. Thank you everyone for today's session. So we will see you soon in 2 weeks. Bye.